Hello students, in previous part we have studied about how the every MacLeod and McCarthy show which one is the genetic material. And in this part we discussed on confirmatory experiment which was proposed by Alfred Hershey and Chase. Experiment is genetic material. This experiment was to show the which substance is genetic material. And the test experiment name is confirmatory experiment. This experiment was proposed by Alfred Hershey and Chase in 1952. They tried to show which one is the genetic material. They selected two substances, two things. First one is T2 phase. T2 phase is bacteriophages, the virus which affects the bacteria. That is called bacteriophages, or we can say that T2 phase. And second one is E. coli bacteria. It means they selected T2 phase, or you can say that bacteriophages. The virus which affect the bacteria, that is called bacteriophage or T2 phase. And second one is E. coli bacteria. We have already discussed about T2 phase, structure of T2 phase in +1 class, but to demonstrate how the structure is performed, a T2 phase bacteria has a polyhedral head. It contains a polyhedral head, a middle piece. This polyhedral head is made up, this is the head, is made up of, it is containing protein and the genetic material DNA. The protein in the T2 phase in neck, it is present in spiral form. And lower end, at the base of the neck, it is this dark line is called end plate. End plate. There is a perforation between the end plate. It means the substance is transferred from this one to other bacteria. It is a perforation. And at the end plate there are legs like structure, this one. There are 6 legs like structures. This is the structure of T2 phase, or we can say that bacteriophage. It contains a polyhedral head which contains protein as well as genetic material DNA. And the neck or middle piece, it contains spiral protein. Just base of the neck or middle piece, there is a perforation through the genetic material. We can say that the material will be transferred from this one to another organism. And at plate there are 6 legs like structure. These legs like structure help in absorption. It means adoption. It sticks with the other organism. It contains protein as well as DNA, genetic material DNA. Protein contains sulfur but not phosphorus, and DNA contains phosphorus but not sulfur. They radioactive both the substances, means protein sulfur means S35 with radioactive substances. They affect the sulfur with radioactive as well as phosphorus P32 with radioactive. Then they show how the process can be accomplished. This process may be completed by the 3 steps The first step is infection. Infection. It means they took different separate experiment in separate vessels. Means T2 phase which contains sulfur read with the radioactive substance, affected with the, or we can say that infected with the E. coli. And second one, phosphorus radioactive P32 with the E. coli in separate. Infection, first of all, the radioactive sulfur and phosphorus. Sulfur means S35 and phosphorus P32 with infected the E. coli bacteria. This is called infection. First of all, the radioactive phosphorus and sulfur contain T2 phase affected with, or you can say that infected with the E. coli bacteria. Second one is blending. As the T2 phase bacteria attach or adsorbs with E. coli, the substances may transfer. Means the genetic material, though the substance which is the genetic material is transferred from perforation site to the E. coli. That is called blending. After that, the mixing of the chemical substance of T2 phase with E. coli bacteria. And third one is centrifugation. Centrifugation, it means the T2 phase bacteria, or you can say bacteriophages, is separated from the E. coli. Then the T2 phase separated from E. coli in different test tube, you can say the vessels. Then they collected some substances such like this one. Means we can say that this is the A, the sulfur contained A protein, or we can say that the phosphorus is B. So different vessels, in vessels A, or we can say that test A and test B, how the reports can be recorded. In test tube A, as we have discussed earlier in +1 class, viruses are lighter than bacteria. So there are the two separate substances may be collected in this test tube. The lighter may be upper side, the heavier towards the lower side. The lighter, this is the T2 phase, remains upper side, and the pellet is bacteria. How is this affected? Suppose this is the E. coli bacteria. This is the E. coli bacteria, first one is affected. This virus is, or T2 phase is the lighter than the bacteria, so it remains upper side, and pellet of bacteria remains lower side. They saw that in supernatant, it means the T2 phase, in test A, which contains sulfur. The supernatant, it means the bacteriophage, it contains radioactivity. And there is no radioactivity in the E. coli. No radioactivity substance can be transferred in E. coli bacteria as they contain sulfur. Sulfur means this is the protein substance. So they saw that the protein is not a genetic material. Then they took another experiment in another vessel, suppose test B. Same activities. The lighter supernatant remains upper side. And the heavier pellets is the pellets which contain bacteriophage, E. coli bacteria. Here they saw that this supernatant is a non-radioactivity, not contain radioactivity, but pellet contains radioactive substance. In supernatant, no radioactivity or radioactivity, but in pellets, in E. coli pellets, they saw that there is radioactive substance is may contain that contains phosphorus, and we know that the amino acid which contains phosphorus, that is DNA, and sulfur that contains protein. Here they proved that only the DNA is the genetic material, not protein, because the radioactive material may transfer from P2 phase to E. coli bacteria, not in protein, because here the sulfur contains. This radioactive substance will transfer from supernatant to the pellets, or we can say that E. coli bacteria through the phosphorus, and we know that the phosphorus present in DNA. So here they prove that DNA is the genetic material. Clear? In next part we will discuss on different types of differences between RNA and DNA.